Good morning friends, let us start the continuation of insulin which has been done in the first part. Now this becomes your second part. Into the first part, what we did is the introduction of insulin, where it is being secreted, synthesized, and in how many forms it has been released, and what is the story of beta islets of Langerhans, where it is being synthesized and stimulated and also enters into your blood circulation. Now today we'll see about the transformation of insulin, how it is being diversifying the content of glucose which is present in your blood circulation to various parts. That means it is diversifying to liver, it is diversifying to your adipose tissue, it is diversifying to your muscle. So these are the three various parts where the glucose has been diversified, converted into different contents of proteins, fats, in the forms of triglycerides, in the forms of glycerol, in the form of glycogen, and in the form of proteins. So we can today we'll see about the story of it, and we know that, and I'm telling you that we have to go to your first part of my video so that you can understand the introduction of insulin. Now we'll the continuation of the insulin, and we see that BLH of Langerhans. Just we have a little recap of it, and we'll just try to go into the next part. And we saw in the first class, it has been synthesized from the BLH of Langerhans, and it is being processed into various forms, because insulin is a precursor. We can't give it into the direct part. It has been available in the form of pre-pro insulin, converted into proinsulin, and it also converted into insulin hormone. And it is done by various parts of your cell, nothing but your rough endoplasmic reticulum, Golgi apparatus, and onto the mitochondria. Until it reaches the mitochondria, it takes the various steps from pre-proinsulin, proinsulin, and the insulin hormone. And into the mitochondria, whenever it is being stimulated, it is releasing a lot of ATP molecules which has been striped up your insulin stimulation, that is from your biliophilia glands, and they are being poured into the circulation. And along with the circulation of insulin, here you can see the receptor of it, where it is being transported into the blood circulation and transported to your blood. And along with the insulin, you can understand that insulin is not only the one which is being released into your blood circulation, it is also entered with the help of one more compound called as C-peptide, nothing but it's a protein part. And this C-peptide is also one of the causes which is causing your diabetic mellitus 2, because insulin and C-peptides are simultaneously being released into the blood circulation. That means they are being balanced with one by other. If at all the C-peptide is less, the insulin is also less. If the C-peptide is more, the insulin is also more. So we can understand that if at all there is lesser percentage of insulin has been entered into the blood circulation, that means your C-peptide is also less entered into your blood circulation. If at all insulin is causing you DM first part, that means diabetic mellitus first or diabetic mellitus 1, and the C-peptide is causing you which is called as diabetic mellitus 2. That means we know that the diabetic mellitus is one of the disease where it is being caused by higher concentrations of the glucose and the lower percentage of insulin is being stimulated. And into the first class, we were just telling you about two hormones which are released in the pancreas of acne. What are they? They are the alpha cells which is releasing your glucagon, and the beta cells which is releasing your insulin. And we know that if at all there is a higher percentage of glucose into the blood circulation, automatically insulin is being released and enters into your blood circulation. If at all the percentage of glucose is lesser, that means we want glucose to be entered once again into the blood circulation, then it is being stimulated by the hormone called glucagon. Now we can understand that one is doing for hyperglycemia and the other one is doing for hypoglycemia. The hyperglycemia is nothing but the higher percentage, nothing but more than 90 milligrams in a blood circulation. It is hypoglycemia. It is less than the 90 milligrams in the blood circulation. So it depends upon the circumstances, depends upon the body stress which we need. It is being stimulated whether what to release, whether to the insulin or the glucagon. Now as we are telling the story of insulin, we can understand that now the condition is hyperglycemia. That means higher of 90 milligrams. Now whenever our body circulation has been traveled with the absorption of the food what we take, that means in the form of carbohydrates, which is nothing but your glucose, enters into your amino acids, which is nothing but your protein part, which is enters into the form of fat, nothing but your glycerol. So these three percentages, whenever your blood circulation is completely full, it has to be diversified to all parts of the body, so that the percentage of the glucose has been lesser in your blood circulation. This is the only first motive that insulin has been stimulated. Now we can understand that your hyperglycemia condition is now, and the insulin is being stimulated, and both the C stimulated, that means C peptide, and also the insulin are being entered into your blood circulation. Now the blood circulation has to be given the answer for the question, that is 19 milligrams, which is being released in the form of carbohydrates. Now we can see in this picture, first have been done with your liver, the second one is your adipose tissue, and the one is your muscle tissue. Now we'll see the story of liver, where what is being done. Now whenever you have your percentage of glucose about 90, that means the insulin came and sat over here. That means the glucose has been stimulated to diversify towards the liver. And here we can see the diagram of here, two receptors have been given, and notice there is the one receptor which is being stimulated for the insulin to collect from the blood circulation. So the liver is having some parts, so that means the receptor, the protein parts of their cell periphery, which has been collecting the insulin part and diversifying towards inside. Whence we can understand over here. And now this part, insulin, which has been collected by the receptor, enters into your liver part, where it is being doing two transactions. That means two way of cycles. The first cycle is, it is being taken up by your, we know that there are receptor parts, what is called pKa and AKT, nothing but phosphokinase 1, and this is phosphokinase being released by your trimers, what is been alpha, beta and gamma, which is been by adenyl cyclase, adenyl cyclase to phosphokinase, and phosphokinase to the genal part, and the genal part your nucleus. That is, the genal part which has been released from your nucleus part is directly stimulating your glucose. And now you can see one more receptor over here. Now here it is indicated by a higher glucose content. That means your blood circulation is in the higher percentage. Now whenever this is being stimulated by this receptor, automatically the glucose which is present in your blood circulation is also entered inside your liver part. Now this pKa or pK1 or AKT, which has been released over there, are being stimulated and giving a striker coin to the glucose to do two functions, as I said before. The first function is glycolysis function. That means whatever the percentage of glucose has been entered into your liver cells, it is being cut down. That means glycolysis. Glyco means glucose, and the lysis. Lysis means cut down. So we can see the words here. Glycolysis is a cycle where the glucose is cut down. That means the percentage of glucose which is present in your blood circulation taken up by the liver, and it is cutting down the glucose. That means it is making the breakdown of the glucose into two parts. One is the glycolysis cycle, which is being transferred into pyruvic acid, and the pyruvic acid to the acetyl coenzyme A, and the acetyl coenzyme to your ATP molecules. And this ATP molecules that are being released enters into your Krebs cycle, and the Krebs cycle does its process and giving you some more content, so that the ATP molecules are being stored up. Now this is the first cycle, as I said before. And in the second cycle, what we can see is the glucose is conformed into or transformed into glycogen. Now glycogen is one of your last part of your glucose, where it has been a stored part. That means the storage conditions of glucose is always in the part of glycogen, not the glucose, because it's having its instability, isn't it? Now the glycogen which has been released from the glucose going on transforming itself and forming into glycogen. Now this cycle is called as glycogenesis. The glycogenesis here, where the word is showing you, or the impact is showing you, that it is glycogenesis. Glyco means the glycogen what it is released, the gene is the generation, and the cis is the process, right? And the glycogenesis is a process where the conversion of glucose is done directly to the glycogen part. Now we can understand here there are two cycles in the liver. One is your glycogenesis, and the other one is glycolysis and the Krebs cycle. These are the three more cycles which can be done in the liver part, where the little percentage of glucose which has been transferred from the blood circulation to the liver. So we can understand that almost 30% has been gone up from the 90 milligrams. That means we can understand that almost 30 milligrams has been diversified to the liver, taking the amount into the two forms of cycles. One is glycolysis, which enters into the Krebs cycle, and the one is called as glycogenesis. Now we'll see the second part, what is called adipose tissue. Now we can understand that almost the glucose has been done over here. Here also it is doing some more conversions or some more transformations of the glucose, and some percentage of insulin has been collected by the adipose tissue. We know that adipose tissue is one of the tissue where the storage content of fat is done in the form of TG. TG is nothing but your triglycerides. The triglycerides are being collected and stored in the form whenever we have a starvation, whenever the body is under stress, whenever the body is under pressure, the triglycerides will come back and give you in the form of glucose. Now here, as the glucose is more into your blood circulation, now that has to be stored in the form of triglycerides, right? And here also the same story runs from the liver and also the adipose tissue. The insulin is being collected by the adipose tissue by a receptor which is present and collecting, which is in the form of PI3KK or AKT, nothing but phosphokinase or phosphotrikinase, right? And here this one is one of the one which is being collected by the adipose tissue and being stimulated by the percentage of glucose enters over here. So the glucose is also being collected by the adipose tissue, the insulin is also being collected. Now this PKT which is being stimulated, and that is being stimulating your glucose to do two more cycles over here also too. What are they? The first cycle, as we understand from the top, that is the glucose is break down. That means it has to broke into different cycles, because the glucose has to be released, it has to be taken from the blood circulation and enters into your adipose tissue. And so the cycles what are being done in the liver is also continuation in your adipose tissue. Nothing but your glycolysis cycle and the Krebs cycle. The glycolysis is nothing but the breakdown of glucose and into pyruvic acid, and the pyruvic acid to acetyl coenzyme A, and the acetyl coenzyme is collected by the Krebs cycle, and at the end of the Krebs cycle it is giving you some energy molecules what are called ATPs. And the second cycle what we can understand here is the receptor which is collecting from the adipose tissue, this is called as GLUT4, and here it is called as GLUT2. That means it is nothing but GLUT means glucose and T is transporter. That means it is having GLUT4. It is nothing but your GLUT4, and this is converted as GLUT2. Now the glucose enters to the GLUT4 and keeps the two cycles. One is called as GP3, which is enters into the glycolysis of pyruvic and the acetyl coenzyme A. And now this acetyl coenzyme A is being collected by the Krebs cycle, convert themselves into succinic acid. We know that cycle of Krebs cycle. And finally we can understand that the ATP molecules are produced over there. And finally, whenever this PKT3 is also produced, it is stimulating one more cycle over here. What is that cycle? A percentage of amino acids which are being released from the blood circulation are having a path towards your adipose tissue, converting your glucose or the taking part of that glucose and converting themselves into DHAP. And now this DHAP is converted into glycerol. And if you can understand that, if you have 3 molecules of glycerol, they are converted into TG. That is nothing but your triglycerides. Now you can see the cycle that the glucose is not only helping your percentage of cutting down your glucose by the insulin and converting themselves into pyruvic acid and the acetyl coenzyme A, but it is also making your that means not amino acids, it is nothing but your fat. The fat is converted into DHAP, and the DHAP is converted into glycerol, and the glycerol is taken up in the form of triglycerides, which is being stored up in the adipose tissue. So the glucose, once again, 30 milligrams is being diversified. You can understand the story here. The first part is 30, the second part is 30. That means the adipose tissue taking the glucose and collecting the fat contents what we taking from the blood circulation, and that means which is being absorbed from the intestine, it is being stored in the form of triglycerides in your adipose tissue. Now the story came back to the last final one, which is nothing but your muscle ones. And here we can see that the insulin which is being produced into the blood circulation, the glucose which is present in your blood circulation they are also diversified towards your muscle, and the muscle is the one which once again consists of both the receptors, that means the insulin collected receptors and also GLUT4 receptors. The GLUT4 receptors is nothing but the collection of glucose diversifying from your blood circulation, and the other one is called the insulin collector, where it is having a receptoral point onto the muscles taken up by the muscle. And here also you can see the two processes continuously. One, the process is glycolysis cycle, Krebs cycle, and the Krebs cycle enters into the form of electron chain transporter, and this electron chain transporter gives you a lot of ATP molecules. So the glucose content is having the first cycle, what is called glycolysis cycle. Now the second cycle is there. What is that? This is nothing but the collection of amino acids which are being absorbed from the intestine. Even the blood circulation is a higher percentage of amino acids. So these amino acids are all diversified towards your muscles. What are the muscles having? The muscles are having one more point, what is called amino acid collector. This is called amino acid reuptake. So what we can understand that the amino acid is being recollected or taken up or uptaken by the muscles, and the glucolysis cycle is being done, enters into Krebs cycle, enters into the electronic chain transporter, which is in the form of ATP molecules. This is the first step. The second step is the collection of amino acids. That means the reuptake receptor or the reuptake transporter. This transporter is collecting all the amino acids from the blood circulation, enters into your muscle part, which is being taken up by the receptor. Where the here you can have to remember the same point what I told you here. One is PKT and the one is AKT. So the PKT is the one which has been stimulating your glucose to diversifying into two cycles. One is the glycolysis cycle and the Krebs cycle, and the other one is nothing but your amino acid uptakeal part. So the amino acids have been taken over here, make up into monomers first. That means the glucose is being collected by the amino acids, a collection, and first they are transferred into monomers. The dots which is being indicated over here are called monomers, and all these monomers of amino acids taking with the help of glucose converted into a chain of protein. That means we can understand that the protein synthesis is being happened. That means the proteins are prepared in your muscles. The ones with the monomers has been taken up, and all the amino acids have been converted into a long chain by the use of ribosomes, and they are being taken up and collected into a protein chain. That means almost 21 to 22 amino acids are collected and form into a protein chain. Now we can understand that the protein what is released over there is used for the repair of the muscle, and also it is being helping for the degeneration of muscles, contractions and relaxation. The glycolysis cycle is supporting you with ATP molecules, and the amino acids are giving you a lot of proteins over there. We know all the functions of proteins, what it does. It does a lot of functions. One is the degeneration of repairs, all the cell repairs, and also it is used for the contractions and relaxations. And the ATP molecules which is being provided over there by the glycolysis cycle are being powerful to give you the energy for the whole muscle to push up their work whatever it is needed. If the body asks you to do some contractions, it do contractions. If the body asks you to do some relaxations, so they do the relaxations. Now this is called as amino uptake. Nothing but you can see the cycle what is called as glycogenesis cycle in the liver, the lipogenesis in the adipose tissue, and the protein synthesis in the muscle tissue. So now we can summarize the part with the 4 to 5 points. The first one is it is increasing your glycogenesis, it is increasing your lipogenesis and increasing your protein synthesis. And the second one is it is reducing the percentage of glucose in the blood circulation, it is reducing usage of amino acids, it is reducing the usage of fat content and stored in the form of triglycerides. And not only this, they are increasing the cycles of glycolysis, they are increasing the cycles of Krebs cycle, and also they are increasing the cycles of DHAP, and it is increasing the formation of glycerol. So altogether we can see that there is insulin diversifying the glucose into various pharmacological actions to do and support so that the blood is being controlled with the percentage of glucose. So this is the story of glucose how it has been diversified into the all parts of the body, so that the content of glucose is being done down into the blood circulation. In the next class, we'll take up the question what is called the preparations of insulin, what are the doses and how we need to take. Thank you, and make my channel for growth, like, share and subscribe, so that my channel will be having some growth to its fun, and it will be an encouraging part so that I can do more videos. Thank you.